University, Uttar Pradesh, Lucknow campus on this international conference come workshop. And thank you very much for accepting our invitations. And I go through your topic, that is neuronal stem cells, which is a contemporary dual tool for disease modeling and therapeutic in regenerative neuroscience and an emerging alternative to animal models. And I definitely assure that your top title is very emphasizing about what you want to tell to our students. And though it is going aligning with today's topic, which we are planning to alternative to animal experiments, with that, I just say very good luck to all my students and other faculty members, those who have joined here in Zoom platform. This session is definitely going to give you some different aspects of alternative tools. Thank you so much once again, Dr. Tavinda, to join us.
Yes.
I can ask Dr. Lucy to please proceed the meeting.
Thank you, ma'am. A very pleasant good afternoon to all the attendees online and offline. And here, now we have arrived at the third technical session of Day 1 AAE 2023. And the honorable speaker for this particular session is Dr. Tavinda Hassan, Associate Professor, Human Anatomy, Histology and Embryology, College of Medicine, Princess Noor bin Abdul Rahman University, the Kingdom of Saudi Arabia. Dr. Hassan is presently working as an associate professor over there, and PNU is an accredited university of the Ministry of Higher Education of the Kingdom of Saudi Arabia and the largest women's university of the world. She pursued her MBBS in 2003 and MD in anatomy in 2006 from Jawaharlal Nehru College of Medicine, Aligarh Muslim University, India. The honorable speaker was awarded with her PhD in anatomy in 2011 from College of Medicine, Swami Vivekananda Suvarti University, Meerut, India. The speaker has been involved in teaching anatomy to medical and allied health sciences undergraduates, including all aspects of human anatomy, such as gross anatomy, oral and maxillofacial anatomy, neuroanatomy, osteology, histology, embryology, and imaging, applied anatomy, and comparative anatomy for more than 10 years, and has been proficient in cadaverics, dissections, prosections, and basic surgical skills. The honorable speaker depicts an extraordinary research profile in the field of medicine with almost 40 journals publication and 428 citations, with H index 13 and item index 14. Recently, her book Anatomy Dissections, The History and Implications of Human Body Butchering, White Falcon Publishing House, has gained high acceptability in this particular arena. She has attended numerous conferences across UK, USA, Europe, Asia, and Middle East as honorable speaker, and she has also handled almost 10 projects in the field of medicine education and 5 projects in the field of morphology, cell development biology, epidemiology, and applied anatomy as supervisor and co-supervisor. So we are really blessed and honored to have you, ma'am, in this particular platform. And I do hope, and I am quite sure that this particular session of yours on neural stem cells, a contemporary dual tool for disease model and therapeutics in regenerative neuroscience and emerging alternative animal models, is definitely going to help our students in this particular field of research. So without further delay, I will request ma'am to kindly present your talk.
Yeah, hello and good afternoon, everyone. I hope I am audible. I would really like to have my video, but let's see.
Ma'am, you are audible and you are also visible to all of us.
Okay, my video is visible.
Yeah, ma'am. You're quite visible
okay.
and audible.
Okay, that's great. So, first of all, I'm very honored to be part of the Amity faculty. And actually, I also have emotional relationships with this university because even my son is studying clinical psychology in Amity. So it's a university, it's a name that's very close to my heart. And without further ado, I will come to the main reason why I chose this topic. So until now, we have been using animals for a lot of researches. And finding a suitable alternative to animal testing is one of the biggest challenges now in the research sector. Because animals, first of all, keeping animal houses is expensive. The second thing is that animals do not in reality exhibit all the plethora of what a human disease is really like. And even because most of the diseases, especially in neuroscience, the diseases of the human brain, they are polygenic, and they have various phenotypes which cannot uh, totally be portrayed by a simple animal model. We have simple animal models. For example, we have zebrafish. Then the closest, as of now, that we can say the closest to humans is the rodents. But still, they are either we do drug testing or we want to observe their behavior. We have different tests that portray anxiety or depression, schizophrenia, Alzheimer's in animals, but we can never be really like 100% sure that this is what it would be like in a human brain. But the problem is we cannot use human subjects. So what is the alternative? Now we have come with ways to kind of grow a brain in a petri dish. So it is a human brain, it's an organelle, it's like a mini-sized brain. So that's what I'm going to talk about today, how we can use embryonic stem cells for experimentation. So they are capable of growing and differentiating into a wide variety of different cell types. They can even grow into a human organ. And through those, we have the ability, a better ability to test the toxicity of a substance, even the behavior, the effect of gene editing. So it's like as close as possible to a first-hand experience that we could have ever had. So coming with this introduction, so let me proceed. So what we will do is we'll just look at a brief history of how stem cells came into existence, but I will talk about it later in my presentation. I will first deal with what are stem cells and how we can use them as an ideal tool for disease modeling, and how we can knock out genes to mimic diseases and also to mimic health. So in part one, we will explore how the neural stem cells, so I'll be focusing especially on the brain, how neural stem cells have become a contemporary dual tool, not only for regenerative medicine, but also for modeling disorders. So as we will go along, we will see the definition of what a neural stem cell is. So I will consider all kinds of audience, maybe people who are new to this field, people who are really pros and experts to this field. So catering to both kinds of audience, we'll first just see what are neural stem cells. We will discuss their origins, and then we will explore how to identify neural stem cells. And we will follow that with exploring what are the resources and different sources of neural stem cells. Then an important section will be the concept of dual use for regenerative medicine and disease modeling as an alternative to animals. So now we are still in that phase when we have animals, and side by side, we are kind of preferring stem cells to animals, but still it needs like very sophisticated labs. So that's another problem why it's still not so common, but we hope to reach there someday. So finally, we will discuss the different sources of neural stem cells. So let's go along. We'll first see what are the stem cells. So let's explore how the stem cells come. Neural stem cells are undifferentiated cells. So they don't really have the phenotype. So it's not a muscle cell yet, it's not a nerve cell, it's not a cardiac cell. So they are undifferentiated, defined by the capacity for self-renewal and multipotency. So during the central nervous system development, the neural stem cells proliferate and divide to generate clonally related progeny that differentiate into neurons. So as you can see in the picture, astrocytes, oligodendrocytes. So these are the different kinds of nerve cells that could come from a stem cell. Now, there are two kinds of division. I'll just cover them in short. The symmetric division, as you can see, the symmetric division of a stem cell underlies their ability to self-renew and serve to maintain the stem cell population. So this is what we look at as a researcher because we want to maintain our population. In contrast, asymmetric mitosis produces one neural stem cell and one neural progenitor cell. So these are the daughter cells with differentiation capacity restricted to neural or glial lineages. So the one thing really to remember about these is that by definition, neural stem cells are defined as cells that have the capacity to self-renew and give rise to all lineages of the central nervous system, namely neurons and the other cells that we know about, astrocytes, oligodendrocytes. Okay. Now, because you will have heard of stem cells in general or in the broader context, it is now important to place our neural stem cells into this wider context and explore their potency. So we have totipotent cells, which are the most versatile of stem cells. So when I say versatile, I mean it can literally grow into any cell of your body. So when a sperm and an egg cell unite, they form one cell-fertilized egg. Now this egg will grow into a whole organism. So we can say at this point that this egg has the capacity to develop into almost any organ. So this cell is a totipotent cell, meaning that it has the potential to give rise to any and all human cells. Now, such as brain, liver, blood, heart cell, it can even give rise to an entire functional organism. The first few cell divisions in embryonic development produce more totipotent cells, but after 4 days of embryonic cell division, the cell begins to specialize into pluripotent stem cells. So that's the second category that I'm talking about. So these pluripotent stem cells are almost like totipotent, but they will not give rise to almost every cell. They will have some limitations. So they can give rise to tissue types, but unlike totipotent cells, they cannot give rise to an entire organism. On the 4th day of development, so we are talking about different stages of embryonic development after fertilization. On the 4th day of development, the embryo forms 2 layers. We have an inner layer and an outer layer. The outer layer is what we call the placenta. And in the inner layer, we have an inner cell mass, which is from where we derive all our stem cells. We harvest all our stem cells for our experiments. So this is something that's really important for a scientist. The inner cell mass can form nearly any human tissue. So as these pluripotent stem cells continue to divide, they begin to specialize further. Moving on now to multipotent stem cells, these are less plastic. So we come to the last category, which is multipotent. They can still form different cells like the brain cells, the heart cells, the blood cells, but still they are limited. So they will form only one type. So if we condition it, if we program it to form brain cells, it will not form heart cells. If we condition it to form heart cells, it will not form blood cells. So to summarize, a little exercise here. Please recall which cell type can give rise to totipotent stem cells, what is an example of pluripotent stem cells and what is an example of multipotent stem cell. So that's just a brain exercise for you. So what we see now, a totipotent stem cell, a good example would be a fertilized egg, let's say up to 4 days. A good example for pluripotent stem cell would be the inner cell mass of a blastocyst. And then finally, neural stem cells, which is the topic of this lecture, are actually multipotent stem cells. So now let's see what are the origins of these neural stem cells. Let's look at the origin of these cells within the developing embryo. So I've tried to really simplify things for you. Neural stem cells are the source of all neuron types in the central and peripheral nervous system. So at the neural stage, there is a neural plate, newly formed neural cells. That is, I'm talking about the embryo. So the newly formed neural cells that are shown in light blue here are flanked laterally by epidermal ectoderm, the notochord cells, which are labeled N. These underlie the midline of the neural plate, and the segmental plate, mesoderm, labeled S, underlie the lateral region of the neural plate. At the neural fold stage, the floor plate cells, they are labeled F here, are evident in the ventral midline, and the somatic mesoderm begins to develop. At the neural tube stage, which is the second diagram that's circled in red, the roof plate I have labeled with R, it's in red color, begins to differentiate at the dorsal midline, and the neural crest cells that I have labeled NC. They start to delineate from the neural tube. So now the process starts occurring. So we see the position starts changing. So then the positional information, so every cell group is encoded and is given a message to go to a certain place and lodge itself in a certain place, because we are now forming the brain. So the positional information allows the neural stem cells to differentiate, proliferate, migrate, and to cover the needs along, like as if in a small mini brain, let's say an organelle, it's not yet a full brain. So they will align themselves as if in a real brain in different directions, anterior, posterior, dorsal, ventral axis of the neural tube. So in this diagram, you will see there are so many transcription factors that are responsible for this that would be, like the topic of an entire other lecture. But this is good for you to be aware that positional specification is taking place. So with these slides, I finally want to bring some clarification on terminology, because we will be using these terms as we go along. As you might read articles, maybe employing the following terminology. Neural stem cells from the neural tube are commonly called neuroepithelial cells, which are the most primitive form of neural stem cells. But in the cortex, the neuroepithelial cells change. They transition into radial glial cells, which is also a form of neural stem cells, and equally important for a researcher. So again, the central nervous system begins as a tube of neuroepithelial cells, the most primitive form of neural stem cells. In the cortex, neuroepithelial cells transition into radial glial cells, as you can see in the picture, and these then give rise to neural progenitors. So now we have the different types of cells that normally are found in the brain, whether it's an animal brain or a human brain. Now they differentiate and they are destined now, they acquire their final phenotype as a neuron, as an astrocyte, as an oligodendrocyte. So in regions of the developing nervous system, such as the spinal cord, the corpus striatum, etc., radial glial cells are not as prominent as progenitor cells, and they emerge from non-radial multipotent neural stem cell populations. This is now what I have shown you here is an extract from a poster. The figure that I'm showing you is an extract from a poster that was published in Nature Neuroscience, and I highly recommend it. It could make a beautiful decoration for your bedroom. For those who are interested, the link I have put later on in the lecture. So moving on to the origin of neural stem cells in context of an adult. So let's talk about adult humans. So once development has ended, neural stem cell populations are very rare and restricted. So the new neurons formation literally stops, except in one or two areas. So let me talk about that. Neural stem cell populations are restricted just to the dentate gyrus of the hippocampus and the subventricular zone of the lateral ventricle, as you can see here. And the same applies to rodents as well. So you have already heard of the concept of adult hippocampal neurogenesis, I guess most of you, if not all of you. So these adult-born neurons are actually derived from adult neural stem cells. So this was the first part. Let's move on to the second part. So how did neuroscience begin incorporating stem cells, and how did it develop into a therapeutics in regenerative medicine? So to identify embryonic neural stem cells is a very controversial topic. So you see here, it's controversial. There is lack of very specific neural stem cell markers, which make it very difficult to demonstrate long-term self-renewal and multipotency. That's one of the problems that we face as researchers. You cannot specifically and confidently 100% say that this is a real stem cell, a neural stem cell with the capacity to regenerate. So there is a solution as well. You use multiple markers. You don't just depend on one marker. So like I said, it's controversial. There is lack of very specific markers, which makes it difficult to demonstrate the long-term capacity of self-renewal and multipotency in vivo. So that's our challenge in vivo. So because we are not always, anything that's done in vivo is superior, is considered superior to in vitro. So that's one challenge that we face. Furthermore, to identify the neural stem cells, one could, the cells could change spatially. So the cells keep changing their position, like I showed you in the previous slide. So according to the neural tube position, and temporally, depending on the time, according to the developmental stage, they would change. So the cell would keep migrating. So like if you are trying to mark it, that's again a confounder for you because it doesn't stay fixed in one place. In vivo, several transcription factors are expressed during early neural cell determination and persist in adult neural stem cells. So some examples I will give you. For example, the SOX genes, such as SOX2, are among the earliest genes expressed ubiquitously in the early neural plate. So this is what you can see, and it's shown with a green fluorescent marker protein. So here, what you see are coronal sections from a chick neural tube during development, and the stage indicated on the top are the stages during the chick development, and at the bottom, you see, is indicated the embryonic stage for the mouse. So you can compare between different animals. You can see that the SOX2 expression in green becomes more and more restricted to the area around the central canal, to just a small pool of cells that are just lining the central canal. So that's what I was talking about, that the expression and the position keeps changing. So let's see what happens. There are also late stage stem cell markers, so some that are apparent in the early stages of development and some that are apparent in the later stages of development. So some of those that are intermediate and late stage markers are, as you can see, there is the nestin protein. That's very, very much used in literature. It's an RNA binding protein. Also, there is one Musashi protein. They appear after SOX2 protein and they persist during development in the adult neuron stem cells. So in this case, we are having mouse embryos highlighting the neural tube, but we have done the same. I'll come later to that part in my lecture. We have done exactly the same in human fetal embryonic cells. And again, there is a lot of ethics involved with that, which we will talk about later. So in this case, we have these embryos highlighting the neural tube, and you can see the Musashi as well as the SOX2 expression. So finally, what is important to remember indeed is that SOX2, nestin, and Musashi, as you can see in the pictures, and they have different colors. SOX2 usually gives a red fluorescence. Nestin gives a green fluorescence. We have DAPI, which gives a blue color. So that's how you can mark and identify and see their population. They can be used as neural stem cell markers. However, be mindful that they can be expressed in restricted progenitors, such as neuronal or glial progenitors. So again, that's confounding. There is no, unfortunately, there is no exclusive antigen that has been identified so far to label neural stem cells. And what is currently used in literature is a combination of positive and negative markers. So you derive your conclusion based on multiple markers. So now, there are a lot of different research articles, and I have tried to put the references down in the bottom right of this lecture so that, like, in case you would want to look it up further. So what we saw, one of the property of stem cells is to divide self-renew. Okay, so is there a possible way to really know that they are dividing and self-renewing? How would you prove it? One way to identify dividing cells is to inject BRDU, which is bromodeoxyuridine. Now, this was a very famous and landmark experiment that was done using the BRDU marker by Bolderini and his team. And back in 2018, if I'm remembering correctly. So this was the first time that it was actually proved that it is possible in the human brain to induce the neurons to multiply. And that gave a lot of hope to the scientific community, because we just, put together two and two, like if we can induce the neurons to multiply, then we can as well, produce healthy neurons. Because what happens in diseases like Alzheimer's, let's say, or dementia, what happens in stroke, the neurons just kind of die out. So how about if we could specifically target areas and just trigger that part of the brain to make new neurons? So like it's like getting immortality, which is a human dream. How about if we could make new brain cells that would replace the diseased and the damaged ones? So the first time it was actually proved in humans is what you are looking here. This is the experiment of Bolderini. And what you see here, those black dots, that's where he proved. So I'll come to that. So what he did was he injected BRDU, which is a marker. It is bromodeoxyuridine, which is an analog of thymidine. All of you must be knowing thymidine, I'm sure. So what happens is it will intercalate in the DNA, because DNA replicates, right? There is transcription, there is translation, and we take in the amino acids. So when you inject a labeled thymidine, which will exhibit a fluorescence, it will be incorporated into any future mitosis, any future DNA which will be formed. So, and BRDU actually is an analog. So it's not the real thymidine, but it will mimic thymidine. So wherever thymidine is required, BRDU will go and replace it. So in this case, in the picture on the top right, you see we have BRDU labeling, which are basically those black dots that you see that have integrated BRDU. So that was revealed we further confirmed it with the anti-BRDU antibody. So there was the reaction that was expected. So it was confirmed that the new cells incorporate the new DNA and actually incorporated BRDU, and it could be proved by the antibody reaction. And the maximum incorporation, if I say in simple words, the maximum neurogenesis, the maximum new cells that were formed, we saw it in the dentate gyrus and the hippocampus. So another way, we could use a lot of different markers. You could use also retrovirus for the same. So you could inject the retrovirus locally into the hippocampus. And that's where the cells will be dividing, and they will integrate the retrovirus, and you could label it with a GFP, a green fluorescent protein. So it will show you, so whatever new cells would be formed, their DNA would be showing you that labeled virus, showing the green. So you can see in the bottom right, you can see how the new neurons can be marked through retroviral labeling. You could quantitatively and as well as qualitatively assess it. So eventually, we want to extract these stem cells as a tool. So that would be the ultimate dream that I was talking about. How about if you could tailor and you could direct and you could customize and you could replace our brain when and where needed with these new cells? So let's talk about how to get it. So that's a challenge. So what are the sources? So if we think about expanding from direct neural stem cell lineage, we can derive neural stem cells at any time during development. So that's the beauty of it. You could get it at any time during development. If you are aware of the stages of development, which I have shown on the right side of my slide, you see that from a unicellular stage, we get multiple cells, and we have different stages like blastocyst, morula, gastrula. I'll not go into all those details. That's beyond the scope of today's topic. But anyhow, as you can see in the picture, now this is how the human development takes place. Also the animals, they follow the same track. So starting from the blastocyst stage or the gastrula stage during development, you can extract these cells from the neural plate even earlier or later. So we finally see that we could extract neural stem cells even from an adult brain. So let's see, there are some other sources. Are you thinking about other sources? Yes, also I'm thinking about that. Let's see what are the other sources. And I'm sure a lot of you already know it has been very much hyped, the bone marrow. So thinking about other sources, it can be generated from non-neural, means not related to the nervous system, but other systems. So there is evidence that we can extract it from the bone marrow. We can also extract it from the blood. So although we need a little bit more research on that, there are few papers showing that bone marrow-derived mesenchymal stem cells and hemopoietic stem cells can even give rise to neural stem cells. Now, this is very interesting that a cell from the bone or a cell from the blood could form a brain in a petri dish. Wow, that's interesting. So adipose stem cells, now that we are still exploring further, even our fat cells, they have been shown to be potential candidates to generate neural stem cells. Wow, so the brain from fat cells, that's interesting. When we think about neural stem cells generated from other cells, we can do that via reprogramming, because always you cannot do it unless you change the signal, the message to that cell of how to divide and what phenotype to acquire. So you have to genetically reprogram it, and I will come to that in the later part of my lecture. So you can induce pluripotent stem cells to take different forms, including neural stem cells, which is the topic for today. So let's see. Okay. So once in a dish, so you see in a dish, what is shown here, you have all the different types of cells. You have a brain cell, you have a epithelial cell, you have a muscle cell, you even have a sperm, you have an egg, you have lymphocytes, monocytes. This is a very nice animation just to show how in a dish you get the complete human body. So in a dish in vitro, we need to control, but now there is something, we need to control proliferation, because always stem cells have a tendency to become teratogenic if it becomes uncontrolled, because that's their quality. They multiply. But how would you put a limit to it? Because if multiplication increasing in numbers beyond what you need, that's already teratogenic, what we call. So we need to control proliferation and differentiation. So we need to arrest them at a certain stage. Neural stem cells self-renewal and differentiation is regulated by a precise temporal sequence of growth factor presentation. So this is interesting. There are factors to control it. Intracellular signaling and transcription factor expression, and that's where we target. We target those transcription factors and we regulate their expression to get what we want. So this is an example, but for certain lineages, we do not yet know the factor to obtain these particular results. So still a lot of it is still a mystery to us. This is obviously a work in progress, and if we want to fully exploit the potential of neural stem cells, we need to understand better how a cell is specialized, how it becomes what it is. Such an understanding more fully, what growth factors and gene regulation is needed exactly. We need to understand better how to control the cell cycle. So I'm talking about humans now. We have done a lot in animals, but still we are lacking a true human understanding in terms of human diseases, in terms of drugs. A lot of drugs have been proved to be not toxic to animals, but when the same, based on those animal researches, when the same drugs are given to humans, they are toxic. At the least, if they are not toxic to the first generation, they might induce certain hidden mutations that might become apparent in the second or the third generation. So that's something to really think about. And that's why we want to move on from animals to humans. But of course, we couldn't sacrifice a human. We couldn't make a human into, an experimental subject. So we have a lot of strict ethical laws. We are no longer in that phase. We had the first, actually the first code of ethics for researchers came after the Nazis. That was the first code that became the first document for the research ethics. So that's what I'm talking about. Before that, humans, prisoners, anybody would be taken for research of any form, and there was absolutely no guidelines. But now we do have. So we are like really tight on what we have to do, and we have what we wish to achieve. So we need, now there are challenges. We need to understand how to control the cycle. We need to know how to control the division. We need to think about growing the cells for long-term and banking. And how ethical is it? You take somebody's cell and you keep it forever. So we have, for example, we have the HeLa cell line. That is from a woman who suffered from cervical cancer, and since it was cancerous, the cells had the potential to multiply. So that's where the HeLa, a lot of you would be aware of the HeLa cell line. The name HeLa comes from her name. So the HeLa cell line is from that woman who died of cervical cancer decades and decades and decades ago, and those cells are still immortal. We know those cells by her name. So that's what I'm talking about. It's important to take consent, you know what, even if you die, your cells will live forever. So these are the things that we need to think about when we do long-term banking. We need to prove long-term stability of the cells. We need to characterize them better, check their genetic stability. So thinking ahead, do we want to transplant these cells? Then we need to evaluate cell-host interaction to think about immunology and transplantation biology. So altogether, we still need to build scientific capacity, such as creating carrier development. So I'm not talking about the career, as in you want to become an engineer or a doctor. Carrier. What are the carriers that will carry those cells into your target site? What would be those pathways. So do we have those training courses? We need to establish infrastructure for that. We need to establish infrastructure for devising those novel cell culture methods. How do we expand the cell line? So until now, a lot of experiment, like I mentioned, is being done on the HeLa cell line. It's still with us. And I think it will be with us for many, many, many more years, thanks to the lady who agreed to donate her cells. So we also need to, verify methods for cell sorting. So how would you sort your cells? So now let's delve a little bit further and let's see how we can exploit it as a tool for research. Okay. So when we say exploit, you want to use something to the fullest of its abilities and capability. So first we use neural stem cells as a tool for research. And often this is not something people think first when we think about neural stem cells. Now, this is a very important tool in our research environment. So first, for example, we have used neural stem cells for fundamental discovery. We just discovered for the sake of discovering. We didn't have anything much in mind, rather than we suddenly got hold of cells that have the capacity to divide, and they can kind of take any shape and form. So we did it just for the sake, you discovered something. We didn't think beyond that, how to use it. But later on, science progresses, we build up one block upon another, we decided to exploit it. So we have used neural stem cells for fundamental discovery. So what happens is we have used human fetal, now this is what I'm talking about. Now, again, lots of ethics involved here. Human fetus hippocampal cells. So now when you talk about human fetus hippocampal cells, means you are taking a human fetus that still doesn't have the right to consent, the right to say yes or no, because it's still a fetus, but still it's human. It will grow into an individual if only you would give it the chance. But then you harvest the brain, means you are killing it, kind of, and you cut the hippocampus and you extract the stem cells from there, because human fetal hippocampus is one of the best sources to derive the healthiest possible stem cells. So again, lots of ethics involved here. We have two schools of thought. Some people are in favor because they want the progress of science, and some people are not in favor because they still consider humanity above science. So it's up to you. It's your call. So we have used human fetal stem cells. We created a neural stem cell line, and with this line, we have identified and validated new genes and microRNA, which are involved in the fundamental mechanism of proliferation and neural differentiation. Now, we have pushed this model even further to be more clinical and more applied. How? We have added to the human hippocampal stem cells different hormones. For example, cortisol, which is a stress hormone. We want to know what stress does to our brain. So what is better than to put directly cortisol to a real live human brain? So we drove it to the maximum. We directly are using drugs, using hormones on a real brain now. So what we did, what stress does to our cells, we can measure different responses. The first thing that we notice, now this is very interesting for all of us, stress decreases neurogenesis. Now, this is really interesting. So that's why we have been hearing all along that stress is harmful. So you can see now to what limit it can be harmful. It really reduces the capacity of your brain to form new cells. So now that's disastrous, because everything is governed by the brain at the end. Also, there is something else that was noticed, that um, uh, the effect of antidepressants was more when the stress levels, in brackets cortisol, because that's the stress hormone. When the stress levels of cortisol was raised, the action, the efficacy of antidepressants was more. So using this neural stem cell model in a dish, we have identified the mode of antidepressants as well. So that's also a huge landmark, because we know antidepressants. We don't know directly how they work. Some people say they work by at the neurotransmitters level. They reduce the uptake of certain neurotransmitters like serotonin, like dopamine, but we don't really know what happens. But now we have been able to prove that actually they promote the formation of new neurons. And that's how they help people overcome depression. A lot of work is going on in this area. So this study, what I'm showing you here, got a lot of attention from media. And actually, it was a nice episode of the BBC radio program. So it was called All in the Mind. And also, I have put a link in the last section of my presentation if any one of you would be interested. So it was like really interesting that everything lies in the mind. Our responses, our physical, mental health, everything. So we can obviously use neural stem cells as a regenerative technology. And when we think about cell grafting and transplants, so let's take another example. Now, in case of spinal cord injury, let's take that as a model. Now, spinal cord injury is really bad, because spinal cord injuries would either leave a person fully or partially paralyzed. At the best, partially paralyzed, but then again, you talk in terms of quality of life, you talk in terms of morbidity. So that can never be the ultimate, you know, marker for quality of life and well-being. So being confined, not being able to move, is a bigger marker than death for when we assess well-being and when we assess quality of life. So that makes spinal cord injury really important. So if we take spinal cord injury as a model, what cells could be transplanted? So they have been actually done. Now, what you see here is that they took from the fetus and they directly transplanted those stem cells, and it worked. It was successful. So stem cells or progenitor cells can be collected from the fetus during different stages of development, like I showed you in my previous slide, either from the inner cell mass of the blastocyst, for example, directly from the brain, directly from a fetal spinal cord, from even their olfactory system, or the umbilical cord of the fetus. Now, if I would express my opinion personally, I would prefer to take from the umbilical cord, because at least you are giving the fetus a chance to come out and live in this world as an individual. Because if you harvest from its spinal cord or from its brain, you are deriving from the fetus the right to be an individual, because it will never survive. It will just be in a lab like one of your mouse or rats. So umbilical cord has its limitations, but it's like so much more ethically comfortable and justifiable for a lot of researchers. It can even, and now we are also harvesting cells from the skin and reprogramming them to become stem cells. So these are what we call the induced pluripotent stem cells. So we induce pluripotency and the behavior, the tendency, the capacity to behave like a stem cell, but actually it's your skin cell. So the cell population can be propagated in cell culture and engineered to produce a molecule of interest, or be restricted to a particular fate before transplanted into the site of injury in the spinal cord. So some of these cells of the fetal nervous origin and umbilical cord could eventually be transplanted directly at the site of the injury. And this is what we call autologous transplantation. Sometimes you could do autologous transplantation or self-transplantation, taking the patient's own cells and transplanting into the site of the injury. Let's now put that in context. Let's compare. what are the pros and the cons? So theoretically, let's say we provide a cell therapy for Parkinson's disease. That's all of us know about Parkinson's. It's one of the biggest challenges we still face today. We still couldn't beat it. We still couldn't finish it. It's still very much there. There are drugs to tackle the problem, but we can't still say that, we have a cure for Parkinsonism. Until now, we can't say that. So the program is that we have a degeneration. Now what happens in Parkinsonism? There is a degeneration of dopaminergic neurons in the substantia nigra. So what is the proposed solution? Let's say we talk about a stem cell model. The proposed solution is that we grab neural stem cells and we make them produce dopamine. So we condition them to become dopaminergic neurons. So that's very interesting. So one of our first consideration in the transportation of neural stem cells, where so in this case, let's say the donor is a fetal brain. So again, cross fingers, the donor is a fetal brain. So we will need to expand the cells in cell culture, first of all, immortalize them because you don't want them to finish, like the HeLa cell line that I talked about earlier. That's an immortal cell line. It was many, many, many years ago, if you want to Google about the HeLa cell line. And until now, it's immortal. So literally, that patient is still living with us. So we could immortalize that fetal brain cell. And then we transplant it into our human subject. Think for a moment about the advantages and the disadvantages of this approach. So one of the advantages is that we only require one-time isolation from the donor tissue. And after that, we can expand for multiple recipients. So you use the same product and you expand and you can, give it to many, many, many people. And fetal cells have, kind of very minimal chances of rejection by the recipient. Another advantage is that they have greater stability and plasticity than adult cells. Now, thinking about this disadvantage. Now, let's see what are the disadvantages. First of all, obviously, there is some ethical issue of accessing fetal tissues. Another question, do we need to immune suppress the patient? Do we need a means for reversing the immortalization? What if at some point you decided, you know what, you have taken enough, you want to finish it now? So there are again, do you have the way to reverse that immortalization? Then finally, is there a risk to the patient? For example, tumors. All the stem cell transplants have teratogenic potential. So all of these disadvantages need to be considered. Something important to be aware of is that such approach of using a fetal brain for donor, so for using a specific part of the fetus and you are expanding it to form stem cells and immortalize them, is currently being used for clinical trials for stroke. So they are already using it for stroke. So at this time, we are in phase 2. So they have passed the safety trial, and it seems to be safe for patient now in phase 2. We are assessing the efficacy of grafting fetal neural stem cells into a patient after stroke. So here, now let's say our second donor would be a blastocyst. So that's one of the stages during embryonic development. We take the inner cell mass from the blastocyst. Then we need to derive embryonic cells from the blastocyst. From the embryonic cell, we need to derive stem cells. So one of the strong advantages is that the embryonic cell line can be used for therapy for other tissues. As we saw, they will not be able to give rise not just to stem cells, but you could literally have anything. You could have a liver, you could have a heart cell. Now, this is a really huge resource if you consider it. Now, another advantage is that we do not need to access fetal tissue because you are just taking it from a blastocyst, which is like it still didn't become a fetus in our human definition. In terms of development, it was just like a bunch of grapes. It was a group of cells. It was not an individual. So technically, you are not harvesting from a human individual. So ethically, you are kind of, saving yourself. So is it more reasonable to access blastocyst tissue? And can you ignore that this is the same blastocyst that if you had let it stay another 1 week, it would have been turned into a fetus? So you are playing with the time. So just 1 week prior or 10 days prior, you say this is not a human, and you harvest it. But what if you had not harvested it? In these 10 days, it would have changed into a human individual. So we are, playing with this, uh, of course, because of the ethical guidelines. So think about it. Is it correct to use blastocysts for this kind of work and experiment to treat patients? So again, you have what are the benefits and what are the losses. So you are also dreaming to treat a lot of people. So do we need to immune suppress those patients? Technically, if you talk to an actual scientist who is working in the lab, this is a multiple cell culture step. Lots and lots of technical difficulties, very time consuming. So let's look at our third option. So all of these options are the options when we are trying to sideline the animals. We are trying to do something that will not require animals, that will not make animals so much mandatory in our labs to come to any conclusion regarding anything scientific. Our third donor, let's say, would be a somatic adult cell. So when you say somatic, it's your own cell, maybe a cell of your skin. So we take the patient's body cells, like I said, skin, special cells called fibroblasts, even your own hair. So we will derive them into those induced pluripotency. So again, you get stem cells from there. And then you could redirect it to make a neural stem cell line. And then you could translate. Again, you come with challenges, you come with advantages, you come with disadvantages. Now, one of the advantages that the donor is genetically identical to the recipient, because you take from the patient's hair or the skin and you transplant into the same patient. So the chances of rejection are minimal. You don't need to immunosuppress the patient because it's his own cell. Another advantage is that you are not really harming the fetal tissue, harvesting a human embryo in any way. So that kind of makes you guilt-free. You go off the radar of ethics in this way. Because of course, this is from the patient's consent, from his own body. You are not really damaging anything. Now, come to the disadvantages. One of the disadvantages is that for this procedure, it needs to be repeated for each patient. So should we consider the economical implication of this procedure? So again, it's a very difficult step. Now, I had, let's say, the fourth. Now, this is something very interesting. Our fourth donor, let's see a condition, hypothetical, of course. It's an adult brain. So a living adult brain, by the way, not a dead adult. So this is a little bit of thinking out of the box. So again, like I said, thinking out of the box. Nothing is ongoing to that extent. Of course, it's too extreme. But as we see that the neural stem cells are present in the adult brain, I said they are present in the hippocampus and the dentate gyrus of the lateral ventricle, the subventricular zone, and all those areas, so which are deep inside the brain. So potentially we could access a donor maybe during a brain biopsy, like that in severe untreatable cases of epilepsy, the focus of the seizures is sometimes taken out. So that's a living brain, and that's the last resort of treatment that they do. Maybe you could use that, but then again, that's already a disease. I mean, that was the brain tissue where the cells were already firing at an abnormal level. So that was actually not healthy from the start. So would you want to use that to treat somebody else? Okay. Let's say we imagine about a brain biopsy and we expand the culture to immortalize and then transplant them to the patient. So here again, the advantage, first of all, is we don't require fetal embryonic tissue. So what are the disadvantages? The potentially big risk to the donor, like how could you imagine taking a piece of the brain? Again, also because you took it from somebody else, you need to immunosuppress your own recipient. You need to think about a way to reverse the immortalization because you don't want a tumor forming there. So you need to suppress the capacity to multiply. So let's see if you have a very good friend, would they be ready to give a part of their brain to you? Is that a potential option? I think no. So again, this is more in our dream still. Now, our fourth donor, our last, let's say our fifth or last approach would be in vivo mobilization of endogenous brain cells. So we already have places in our brain where cells are already being formed. How about if we stimulate them a little bit more? We inject those endogenous brain areas with growth factors because they are already designed to form new cells. So the brain naturally harbors neural stem cells. So it has the ability to multiply. So how about if we trigger it? We inject a growth factor intracerebrally inside the brain so that it could mobilize these endogenous neural stem cells. Now think about the advantages and the disadvantages of this approach. Now, one key advantage is that it's within your own brain. You don't require to access any tissue from outside. No ethical problem, no problem of immune rejection. Now let's look at the disadvantages. Now, we have only very limited niches that can make new cells in our brain naturally in an adult brain. And like I had said many times before, one of them is the dentate gyrus of the hippocampus, and one of them is the subventricular zone of the lateral ventricle. So they might have a very limited response. We don't know because even the niche is so small, we don't know how many cells it would end up producing. And even if those new cells would incorporate into the functional brain circuitry, like any new cell formed is useless unless it incorporates and becomes a part of the brain's functional system. So let's say in case of Parkinson's, can those cells migrate as far as the midbrain, the substantia nigra, and produce dopamine? You know, that place where the dopaminergic neurons were damaged in the first place in a Parkinson's patient. Will they even ever differentiate into dopaminergic neurons? So this is an approach that is currently being taken in context of Parkinson's disease. Clinical trials are going on. Patients are receiving injections of growth factors to stimulate their own brain to produce dopaminergic neurons. We are still waiting the outcome. So thank you very much for this lecture. And there are a list of references. I will post my lecture. So please, if you are interested, just go through these. And since I'm running short of time, I will skip. I had to talk about CRISPR technology, but I will just give it a slip here. I will come to the last part, which I feel is worth mentioning since I mentioned it before. A little bit about the fundamentals of tissue culture. So since we started our talk with that, I want to delve just a little bit more into the fundamentals of tissue culture in vitro model systems. So let's see the people who did it. Now, we talked a lot about stem cells, a major advancement in the development of cell lines that can be grown indefinitely and yet retain a normal karyotype. So when we talk about the human karyotype, the normal, and the capacity to differentiate into special cell types. So the isolation of embryonic stem cells initially, so let's look a little bit in the history. That's what I mentioned when I started the lecture, that we will look at the history, how it all happened. So initially, the first candidates were mouse. So it was isolated from mouse blastocysts by Martins Evans in 1982. So I have put a picture of him here. You can see the gentleman here. These cells can, in principle, generate any cell type of the body in a culture dish. So in 1998, this was also achieved by Jamie Thompson. Now, this gentleman On the other side, you see lower down, Jamie Thomson, 1998. Now, this was the first landmark that was achieved for humans because he managed to do the same with human embryonic stem cells and allowed for generation of inaccessible cell types such as neurons and in large numbers for the first time. There are ethical issues, like I mentioned, surrounding the generation of human embryonic stem cells. However, because they require the use and destruction of human embryos. So, but our first landmark, that the name that we still remember, is Mr. Jamie Thomson. Now let's look at the other landmark. Now again, following the steps of Jamie Thomson, he already showed that it's possible to multiply the cells, kind of get a brain in a petri dish. Now, Mr. Yamanka, he went a step further. So following on from the development of human-induced pluripotent stem cells is the exciting prospect of using these cells for personalized and regenerative medicine. In principle, an individual who is suffering from a disease such as Parkinson's, even Alzheimer's, even motor neuron disease, they could give a small sample of blood, which could be reprogrammed into induced pluripotency and then differentiated into, let's say, dopaminergic neurons. These dopaminergic neurons could then be transplanted back into that individual to replace the ones that have been lost. So we already discussed about it. Now to generate these so-called induced pluripotent stem cells to study genetic diseases, this was done for the first time by Yamanka. And he actually discovered factors that were called Yamanka factors, that he said that these are the factors that are absolutely crucial to bring on the induced pluripotency. So there were like 20 factors that he studied, and out of 20, as far as I remember, there were around 12 that he said, or 10 that he said that these are absolutely crucial. Without these factors, there can never be any induced pluripotency. So we still call them the Yamanka factors. So to what you see here, you can see how the neural, now this is what the neural stem cells look like. So they look like small flowers. We call them neural rosettes. That's why we call them rosettes. So you can see the pictures, the iPS cells growing in culture. And once you have stained it with the GFP, the fluorescent protein, you see that's the real shape of the rosettes. And you can stain them with different markers. So neural rosettes are not just the neurons, they are also radial glial cells, and they generate progenitors, which can in turn develop into neurons. And in this particular picture in the middle, the neurons are stained blue. So these rosettes could be considered a kind of a 2D culture model of cortical neural tube development. So here you see that's how, following on from the development of human-induced pluripotent stem cells, it's an exciting prospect of using these cells in personalized regenerative medicine. I talked about a lot of diseases. So you get those cells, somatic cells, the most harmless, you get from the body, the easiest to get from a skin or a hair or even a piece of nail. You reprogram it, you stimulate it to differentiate, to multiply, and then you get any cell that you want. And these transplanted cells will be genetically identical to the individual. So this eliminates the risk of rejection and the overuse of immunosuppressive drugs. So here I come to the end of my lecture. I hope it was interesting. It's a little bit offbeat topic and it's not a very common area. So, but still, I hope I was able to give at least some idea of what our prospects in future hold in the form of stem cells. We could, to a certain extent, do away from animals. It would reduce our costs once we establish the setup for a stem cell. That initially would be costly, but later on it would be almost free for us, like an inexhaustible resource, which is not possible in case of animals. So yeah, I'm open to questions if you want. I'm sorry I took more time than I was supposed to take.
No, no, ma'am, no need of saying sorry. We are really privileged enough to have such an elaborate and informative session from you, and it is not at all offbeat, ma'am. It's quite very relevant to our topic, and it's a very recent and noble work or noble arena, rather, I would say that. I mean, the utility or the involvement or utilizing stem cells now for their medicine, medicinal purposes. I have some couple of questions from our online and offline students. So if you will allow, should I pick some
Okay, sure.
questions from you, ma'am?
Yeah, sure.
Thank you, ma'am. I do hope that this particular session will be of great benefits to our M Pharmacy and PhD students for their research purposes. The first question that is here is from Mohammed Jed Sheikh, and he's asking that how CNS begin neuroepithelial cells.
Please, can you repeat the question?
Hope I'm clearly audible to you, ma'am.
Yeah, you are. Just you could go a bit slow. Yeah.
He's asking that how CNS begin neuroepithelial cells.
Okay. So I hope I get the question right. So what he wants to ask is how, I will rephrase the question, how the neuroepithelial cells develop into the central nervous system. So actually what happens is that during the embryonic stage, the embryo, as we know, as a little human, a little human with every organ, initially it's not that embryo. It's just like like a plate, a disc, like a sandwich. It's a sandwich with 3 layers. So there are these 3 layers, which we call the trilaminar disc stage. And this disc later rolls on and it forms like a circular tube. So you can imagine holding a piece of paper that's flat initially, and that's what you call the neuroepithelial cell disc. And then you roll that paper into a tube, and that's what becomes the neural tube. And now this neural tube will expand in different areas. At the top, it will expand to form the brain. And lower down, it will not expand so much. It will still retain its tubular shape, which will be your future spinal cord. So that's how the neuroepithelium that is coming from the embryonic disc changes into the neural tube, and the neural tube changes its shape and its volume to form your future CNS, the central nervous system, which includes your cerebrum, your cerebellum, and your spinal cord. I hope I answered the question correctly. If I got your question correctly.
That's true, ma'am, and I do hope that you have got the answer. The next question again is from him only, Chair Singh, and he's asking that is there any way out to control proliferation?
Is there
any way out to control proliferation?
Is there? Please, could you be a little bit?
How can we control proliferation?
How can we control proliferation? So this is what I have been discussing all along. It's easy to induce proliferation, but for controlling proliferation, that's our problem until now. When we talk about stem cells, see, until now, I would like to say something. As I study science more, I became more of a believer in a higher power, because we have everything inside us and it is being controlled. Otherwise, all of us, even right from the fetal stage, we would be having a lot of tumors. Even like maybe my brain or my bone or my muscle, my heart, it knows how much to grow and when to stop. Now, the interesting thing here is that until now, we don't know the factors that stop the proliferation, that tell our body, that give us a command that, you know what, it's enough. Stop now. Stop multiplying now. We have it inside us. Now, our challenge, we got hold of the cells that can multiply, and we are still struggling to find factors that will inhibit or stop the proliferation. We do have a lot of factors now that we found, we discovered, but we didn't make it, by the way. They were there within us. So somebody, maybe, you know, if you are a believer, I will say God put it inside us. We are still decoding those. We are trying to identify, okay, so this is the one that will stop proliferation. So we have factor A, factor B, factor C, but did you make it? No, you didn't make it. You just identified it in your own body and you separated it, and then you tried it and you discovered, oh wow, so this can stop proliferation. So that's what I said. I became more of a believer. The more I study science, I believe more. So when coming back to your question, how can you stop proliferation? Yes, we have identified a couple of factors that will arrest the transcription or that will arrest the translation. So without transcription and translation, there can be no multiplication. We have, but then, like I said, we are still struggling. We don't even have hold of all the factors that already reside within us. We have not even been able to identify it. So this is, I hope I answered your question and also gave you a food for thought, maybe like, wow, we have it and we don't know it. We are still trying to identify it and use it.
Yeah, thank you, ma'am. The next question is from Vishal Gyan Chandani, and he wants to know that can stem cell therapy be used in triple negative breast cancer?
Can you just be a bit slow about the question because I have echo?
Sure. Can stem cell therapy be used in triple negative breast cancer?
First of all, see, breast cancer, again, if I break down this question to the very basics. Now, breast cancer, or for that matter, any cancer, cervical cancer, whatever cancer, what goes wrong in a cancer? Cells start multiplying in an uncontrolled manner because somewhere down the line, they lose that signal that was telling them to stop. This is what I discussed in my previous answer as well, that within all of us, there are certain factors that give us the command, okay, grow and you know, repair your body, but don't make a tumor. So coming to your question, breast cancer is what? When the cells multiply and they lose that command, that message that tells them to stop, and there you get a tumor, there you get a cancer that will creep into the lymphatic system and metastasize to different organs, different parts of your body. So how, now this is again something that's hypothetical, it's still not been tried successfully, but yes, you have given a very good idea. Stem cells that could be directed, targeted to reach that tumor site and, you know, kind of have those kind of antibodies to identify the cancer cells. And, you know, those cells could be programmed like our immune system to kill those cells. And also at the same time, give them a message to not multiply anymore. Maybe cells that could destroy the blood supply around that tumor, because no tumor can grow without angiogenesis. Any tumor, before it starts, it creates a platform for it with a very rich blood supply. So it generates, stimulates the formation of new vascular supply, blood vessels that we call angiogenesis. This is a very good idea, but unfortunately, it could be possible, hypothetically, maybe in future, maybe within a few years, we would even be having those cells that could be taken as, you know, soldiers to treat cancer. They would go and just arrest the growth of the cancer cells. But for now, we are still in, like, you know, it's still a hypothetical.
Ma'am, one few more questions to go if you allow us.
Sure, no problem.
Sure. The next question is from Syed Tahir Hussain, and he wants to know that how it is practically possible to develop neural cells from embryonic state. I hope that the answer has been already given by Honorable Ma'am, like the neural tube and all discussed.
Yeah, but I will just simplify and maybe give a one-word answer.
Sure.
See, you have, after the fertilization, when the egg and the sperm meets, what you have is something like a grape, a bunch of grapes. That's not a human still. Now, this bunch of grapes that would later be called technically a human embryo. That bunch of grapes is what you take out. I will be very simple about it. You take it out with a syringe and you get certain cells. And that's from where you get your stem cells, and you will get the brain in a petri dish, like I said, in a plate. You could even make a liver from it. You could make a heart from it. You could make anything from that bunch of grapes. That we call the blastocyst, but I will say bunch of grapes just to, let you imagine how it looks like.
Ma'am, the next question or query is from my side itself. I want to
Okay.
I want to just know that is there any way out that even we can target the glial cells in neurodegenerative disorders, and if so, then what are the possible prime targets that can be targeted for evaluating or for discovering new drugs?
You mean to say that our adult brain
Yes.
cells?
Apart from the regular neuronal cells, like we have been targeting different
Yeah.
markers, the neurons right now, when
Yes.
we are going to certain type of degenerative disorders like Alzheimer's,
Yes.
things like those. But can we target even the glial cells, because they're also the part of CNS? And I do
Yeah.
hope that there must be some sort of deformities even going on in the glial cells that might be contributing to the neurodegenerative disorder. So is that
Yeah.
way out?
Yes, it is very much possible. So look, like in the slide that's already on the screen, it's actually being done. So any cell from your body, I think my screen is still there. Even if it's not there, it's okay. I will just say, to make it like very simple, we want to use the adult cells. So let's say we take hair, we take nail, and most commonly what we are taking is the skin cells, and we take those cells. Now you have cell and you induce certain factors, which earlier I called Yamanka factors. So what you have, let's say, very simple, you have a skin cell. Now you induce certain factors that will reprogram the cell and make it divide, and not only divide into one cell type, but you induce pluripotency. So now this is the skin cell that has the potential according to the message that you are giving it. It can form brain, it can form liver, it can form kidney. And now what you want to do is you want to test for a drug. So what you have as of now is a healthy kidney, a healthy brain, a healthy liver. So I'm talking only about 3 organs. Now, what you do is by certain gene editing technologies like CRISPR, which I skipped, I was going to talk about it because this is exactly what would answer your question. The CRISPR technology is gene editing. So you edit the gene and you induce a mutation that will cause a disease. So you could induce a mutation that would later mimic, let's say, chronic renal failure. You could induce a mutation that could mimic, let's say, cirrhosis or hepatic cancer. You could induce mutation to mimic Alzheimer's by forcing it to form those amyloid plaques. So now you have one group of diseased organelles in plates. I will be very simple. And you have your other control group, which is it's healthy. And now you want to test multiple drugs. So you could have 5 or 6 sets of uh, the disease versus the healthy, and you can test 6 drugs at a time and see how it progresses. Does it progress? Does your intervention, it could be a herbal product, it could be a chemical drug, it could be a hormone, it could be anything. Now, even environmental conditions, maybe you would want to just expose it to natural environment, the things that are so-called very good for us, like oxygen and sunlight, those things, even ecological. So you expose them to whatever you want and see, is it reducing the condition, is it pausing it in any way, or is it like, it doesn't make a difference at all, or is it triggering it in any way? So in that way you would end up with certain uh, products that you can say, to a safe extent, with still cross fingers, you could say, you know what, this appears to be protective. This appears to have a certain degree of intervention, let's say. This appears to be, really harmful. It triggers, it speeds up the condition into a full-blown disease picture. So that's how I hope I answered your question.
Sure, ma'am. One last basic question that if the stem cells are being derived from the first degree or the first generation relative or the siblings, can they when used I mean the sister or brother in that we can exchange it, or is it particular to that particular individual from whom the stem cells have been derived?
If I understood your question correctly, what you mean to say is that when we derive, or when, let's say, when we extract or harvest stem cells, where would be the minimal chances for immune rejection?
Yes.
So the minimal chances would be where with the person with which you share. Now I will go stepwise. The person or the individual with who you share 100% of your DNA, which is you yourself, or the person with who you share 50% of your DNA, which is your father or mother, or the person with who you share 25% of your DNA, which is your sibling, your brother or sister. So it goes in that way.
Okay. Okay,
Yeah.
ma'am. So we are done with almost all the questions
Okay.
in the chat box and the question answer sessions, but still some questions are being floating and due to constraint of time. So with your due permission, if you allow us, we'll share your mail ID to these attendees and they can.
Please, for sure.
Sure, ma'am.
For sure. I have given my mail ID to Dr. Yasir. I really want to thank him to give me this platform. He is a good friend of mine, and I'm very thankful for him because this is a very good event. It was very interesting.
I would request Dr. Yasir to join us here, sir.
Yes. So I have attended a lot of international conferences as well, but the way this one is conducted, very interesting. I heard the earlier talks also. It is very good, and I would also want to be a part of it in future. Like, it was such a good experience for me.
Ma'am, that's so nice of you, and I'm so extremely thankful for you for the words of appreciation. That really means a lot to you, a lot for us, because
Thank you.
because we have been working day and night. All our team members, the student volunteers, the faculty members, our HOI ma'am, the support over here. So we have been looking forward to video of this particular platform, and we'll be really glad enough that if you're planning somewhere, some time of the year to be here as person at Lucknow,
Sure.
definitely have a talk with you, interaction session. We'll arrange with our students. We'll definitely.
For sure, because I keep visiting India frequently, and if Dr. Yasser, he will just let me know, I will plan my visit according to that. I'm really, really waiting to meet all of you in person. It's very well conducted. Very, very well conducted workshop. Hello.
Good afternoon, ma'am.
Good afternoon.
Have a small interaction with the author.
I want to thank you for giving me this opportunity, This was a very good experience for me.
Okay, thank you, ma'am. Thank you very much for your presentation.
And thank you for joining us online and taking time out of your stringent schedule and be here with us, dispatching such a nice, informative, and elaborated session. Your slides were so informative, and I know that how much pain you have taken to prepare. It's all detailed information that have been included, and you have not given a single scope to ask. Put questions, more questions. I'm so happy to find you as a speaker.
Thank you.
International speaker for today's session. So again, our heartiest congratulations to you, ma'am, and we look forward to meet you as a person in the campus.
For sure.
Dr. Lucy.
Yes,
I just congratulate behalf of all our fraternity of AIP. Dr. Tabinder, thank you so much and congratulations for such a nice presentations, and really it enlighten our students. I go through the messages. A lot of them are just love your talk, and I wish you that to Amity Institute of Pharmacy. And thank you so much.
Thank you. Thank you for this opportunity. Have a good day ahead. And I will try to answer all. If they have any questions, even in the chat box, I will answer. I will go through the chat box and I will answer.
Thank
And
you.
my email also is with you if anybody wants to contact me through email. Thanks a lot.
Sure, ma'am.
So have a good day ahead.
Thank you, ma'am. Have a
Thank
good day too. Thank you.
Okay.
So
Thank
with that, we have arrived at the end of today's sessions. That all the technical sessions we have completed, and with that, we have arrived at the end of day one, AAE 2023. So now I request all the attendees to please click and submit their feedback for the first day of international conference come workshop. The feedback link has been shared in the chat box and it will be closed before 5 p.m. today. So it's stringent, it's a strict instructions to all the attendees online and offline. Please go through the feedback form and submit the same before 5 p.m. today, because after that it will be closed. So please fill the feedback form and submit the same before 5 p.m. The same has been shared in the chat box. So now I request that everyone, offline and online, can leave the meeting. And I also request you all to kindly join tomorrow by 10 a.m. because we are going to start our day 2 AAE with the presentation from international speaker, Dr. Nrsinga Misra sir, and it will be definitely started sharp at 10 a.m. So to avoid delaying, please do join on time. So thank you all and have a great day. Thank you.